Hey, how's it going, class? This is Mr. Miles coming at you with a podcast. In today's podcast, what we're going to be talking about fermentation. Okay, so you remember from earlier when we took a test on cellular respiration. We know that cellular respiration breaks down to three parts. It breaks down to glycolysis. It will then go into the Krebs cycle, and then the electron transport chain. Now, glycolysis makes some ATP, so it's a little bit of ATP. Krebs cycle will make a little bit more. But really what we care about is we care about making this NADH and FADH2. And the important thing about both these are what's on the end here, these high energy electron carriers. So these are high energy electrons. When they get fed into the electron transport chain, they can simply move down the electron transport chain, transporting hydrogen ions across a gradient, if you remember. And as these are transported across a gradient, they will then naturally want to flow back up to where there isn't a lot through ATP synthase, making a lot of ATP. Now, you can see that these pumps pulling the hydrogen ions down here are very important, otherwise they wouldn't want to cycle back through ATP synthase, creating a lot of ATP, 32 ATP, to be exact, per glucose molecule. So the thing is, you have to understand is that whenever these hydrogen ions are being pulled across, it's active transport, so it requires energy. This energy is donated to them by these high energy electrons. Now, these high energy electrons will eventually run out of energy as they're drained, giving it to these protein pumps, and hydrogen ions that are left over and O2 will come in, bind with those, so we combine all those, and as you remember, these will all create H2O, which can leave the organism. Now, sometimes we don't have this guy coming in. We don't have a lot of oxygen. We don't either have enough or we don't have any oxygen coming in, or you're an organism that doesn't use oxygen. Well, in that case, you're going to use a method called fermentation. And in fermentation, we're only going to care about the first step. We're only going to care about glycolysis. Because as you remember, glycolysis can make a little bit of ATP. In particular, we can make 2 ATP. Now, that's not enough for long sustained activity in larger organisms, but some smaller ones do just fine. So let's talk about a small organism that can do that. So the first one is yeast. Now, yeast is particularly awesome because it allows us for having bread, and for another thing that none of you should be dealing with because you are all very young, is alcohol. But at the same time is you do deal with it when you have rubbing alcohol or when you have to sterilize things. So regardless, yeast is an extraordinarily important organism in our life. Now, yeast is actually a living organism. It is a fungus. So it's a fungi, and if you remember, fungi is a type of eukaryote. So this small little unicellular organism performs fermentation. And so if you look down here at my chart, we want to pay more attention to this side on the left than we do on the right side just yet. So we can see that inside the cytoplasm, so in glycolysis, the first one, we're going to go ahead and we're going to split a glucose molecule. So this yeast organism will split a glucose molecule. It'll get a little bit of ATP, enough ATP for something as small as yeast in order to function. At the same time, as remember, when it splits that ATP molecule, you're also going to attach a high energy electron onto the NAD+. This poses a problem because once we start creating a lot of ATP and putting high energy electrons on NAD+, you are then very quickly going to run out of NAD+. There's nothing that can get rid of this high energy electron because it doesn't go through the Krebs cycle and the electron transport chain, which will eventually pull that off. So how do we get rid of that? Well, what these organisms do is they simply will rip off that high energy electron themselves and they will throw it onto a molecule of ethanol alcohol. So you can see here that the glucose got split in half, so we have 2 pyruvic acids, and then what it'll do is it'll cut off this CO2 and this CO2, this carbon and this carbon, making 2 CO2 molecules. So you can see the carbon right here and then oxygen molecules attaching. It will then take this CO2 molecules, it'll take these two and it'll kick it out of the cell, and then what you'll have here is just 2 ethanol alcohol molecules, which it can also get rid of. So that's how you create alcohol using yeast. They just generate ethanol alcohol by themselves during fermentation. At the same time as you also get some CO2 gas that's released. Now, this is extraordinarily useful in making bread rise because as you put the ethanol alcohol into there and the CO2 will cause bubbles inside of the bread, causing it to rise, and the ethyl alcohol will actually burn off through the cooking process. And this is extraordinarily important because this accepts that extra electron, so it can cycle right back around and get a new NAD plus to run through the system again, creating 2 ATP. Now, at the same time, is you have other organisms like humans, just in case you were wondering, and also bacteria, but we're going to focus on humans. We can also do fermentation. So let's take a runner. So somebody likes to run. Now, we have enough ATP stored up inside of our cells to last just actually a couple seconds. We actually run out of it very quickly. Well, we're constantly performing cellular respiration to replenish these ATP supplies, and for most activity, we're just fine doing it. However, whenever you do any sort of athletic activity, like sprinting, you're going to run out of it very quickly, and your body is not equipped to make that ATP fast enough. So sometimes what it'll do is it'll switch to fermentation, which it can use temporarily. So let's say you're a runner, you're running like 200 or 300 meters. Usually you're able to do fermentation well enough to last that long. So we're in a dead sprint. The first few seconds we run out of ATP, and we're not able to take in oxygen fast enough in order to run cellular respiration. So our body switches to fermentation. And so it's going to be taking that ADP, creating 2 ATP, and it's going to be taking that NAD plus and making NADH. Now, again, we have to get rid of this high energy electron. So what we do, so you can see that it's showing the NADH down here. So we're going to take that high energy electron and we're going to donate it to the pyruvic acid to create something called lactic acid. So pyruvic acid will become lactic acid when you add an electron to it, especially this high energy electron that comes from NADH. So you're going to get 2 lactic acids. The NAD plus can then cycle back and create more ATP, except the electron that comes off the glucose. So essentially what you have is you have a buildup of lactic acid. Now this can be removed using O2, but again, we're not getting it as fast as we're making it. So after a 200 or 300 meter run, you are going to start breathing very heavily. And the reason why we breathe so heavily is because of the fact that our body's trying to get rid of that lactic acid and bring enough oxygen in so it can perform all the steps of cellular respiration and not just be stopping at glycolysis, so it can go through the Krebs cycle and through the electron transport chain. You need a lot of oxygen, and you have to get a lot of that back in because we went into a deficit because of all this lactic acid that's put into there. Now, the reason why we also get a burning and cramping sensation afterwards is because, yes, this is an acid, and so it does give us those sensations. So all the effects of after running is our body's simply trying to switch away from using fermentation and going right back into normal cellular respiration, and it has to take up this lactic acid that we built up during that time period. So that's essentially what's happening. So yeah, it's a pretty good process, but again, it can't sustain us for very long. And if you're yeast, it's a really good process to sustain yourself for a very long time because you are unicellular, you are very small, and it's great for us humans that yeast does this because we can get bread, and we can also use rubbing alcohol to sterilize things. So this ethanol alcohol is extraordinarily important. So that's fermentation in a nutshell. It's either you're going to make ethanol alcohol and CO2 if you're like a yeast cell, or you're going to make lactic acid if you are a human. And this is all because we have to get this high energy electron taken off of NADH, and one of these 2 things gets created when we do that. So again, if you have any questions or comments or whatever, just feel free to email me or just talk to me in class. And until next time, I will talk to you guys later. All right, bye.